she came from Macedonia 12 years ago. That's a while. And she started at the Roskilde University. She did chemistry and molecular biology. Then a master's in Roskilde, KU, DTU, and now she works for ProDiagnose. I almost went right. PreDiagnose. And she does research in bacterial infections and antibiotics, so she will be telling us a little bit about that.
So thanks for the intro, and I tried to kind of make the topic as broad as possible so we all just get a clear understanding of something new, which is called bacterial persistence. But I'll give you basics, and hopefully I apologize in advance for any busy slides, or you can just cut me and ask me questions afterwards. So I chose the title of Persisting Bacterial Infections because I would like to focus on those infections that are persistent. So it's not something that you take antibiotic for a week and then the infection is cleared. It's something that for people, you can, like urinary tract infections, you get the infections once, after a month you get it again, after a treatment you get it again. Then you have immunocompromised patients that have infections for 10 years or something. So these infections are very bad and they give them bad lifestyle and so forth. So it's just, I'm going to tell you about how, as scientists, do we look at this, and is it important, is it clinically not important, do we see it as often? So in basically this picture just came out and I was really happy to see it because it shows a scenario where once you have a bacterial infections, you take a Superman drug that will basically, it's aimed to kill your bacteria. And then we have our immunity that also takes care for most parts of the bacteria. But once you get something like resistant bacteria, some that are drug tolerant, or some that sleep through the treatment and then they wake up after treatment, it's gone and give you again an infections are called persisters. And these two, tolerant and persisters, are not commonly known. So you guys probably know more about resistance. So I will give you, of course, a little bit of that. So the research area is bacterial microbiology, and I took this slide from my PhD defense 5 years ago. And at that time, there was a paper that came out that stated that we have 10 bacteria per human cell. And that's not true. So a year ago, they figured out that the lab work was bad. So basically, we have 1 bacteria per 1 human cell. And how much is that? Do you know how many human cells we have in the body? Anybody?
A lot.
Yes, a lot. So it's 37 trillion human cells that we have. So even though we have these bacteria, we live in harmony with them, right? But then often they cause disease. And those that cause disease are the pathogenic bacteria. And where can we find them? We find them everywhere. We find them probably not here, but we find them outside in parks, festivals. We find them in the food we eat, hopefully not, but we do unfortunately get some from time to time, from water we drink that is set out. And these bacteria, even though we have the immune system, as I said, they evolve fast. So this video here, it is very fast, and that kind of is the point. So bacteria divides binary, so 1 makes 2, 2 make 4, and so forth. And they divide, so if I give you an example of Escherichia coli or Pseudomonas, they would divide every 15 to 20 minutes. And human cell divides every 24 hours. So you can see how fast, once you get a pathogenic bacteria, it can divide, it can spread, and it can spread to other people. So that's why we get all these alarming news from time to time that tell us, oh, we got this patient in this country and we have no antibiotic because it's a pathogenic, it's a resistant bacteria. And when these alarming news come out, everybody is shocked, like, oh, how come we don't have antibiotics? And we don't, because antibiotics are not blockbuster drugs, so they're not drugs that you take for blood pressure or for diabetes. So companies do not want to invest, so there's no incentive of investing into antibiotic development. So those that we have, we're gonna run out. Hopefully not, but these infectious diseases that kill so many millions is just statistics that I took out from the World Health Organization press releases. They are kind of scary because this pathogen, these infections leads to a lot of deaths annually, and it's still a number which is less than what a cancer the death from cancer is caused. But still, in terms of the state budget, I would say, it costs a lot to have patients in the hospital, and when they're infected with the pathogenic bacteria that you don't have antibiotic for, then you have these patients for an increased time in the hospital, and this leads to an increased cost. So antibiotics are the essential cure of bacterial infections, and this figure illustrates very clearly that how long after you discover an antibiotic you get or you isolate the first bug which is resistant to that antibiotic. So an example here is tetracycline, discovered in 1950, and already in 1959 there is a first tetracycline resistant Shigella superbug that had been isolated. Another example which is concerning, it's methicillin. It got out 1960s, and then 1962 you have the first bacteria. And this goes on, right? So if you rediscover more antibiotics, it's inevitable that you will fish out antibiotic-resistant bacteria. And this is so concerning that in the 2014 report there is this scary sentence that every scientist is using to get more money for their research, and that's that the post-antibiotic era is coming, in which it's not a fantasy that we don't have basically antibiotics to cure infections anymore. It's the same problem now. I went and I checked what's the status report, so it's kind of an ongoing battle. So now you're very familiar with this, and I think many of you are, but I would like to introduce kind of a paradox that is really, when it comes to what's happening in the clinic, this is the paradox. And that's that we have antibiotics which are in fact effective most of the time if you don't have a superbug, but they are unable to cure the infection. Okay, so where is that? The example is these 3 patients I worked with during my postdoc work at the Risk Hospital, in the Central Hospital here at Copenhagen. And what we see now, so this is years of how long the patients have been infected with Pseudomonas, that's the bacteria that I worked with. And you see from 2007 to 2012, so we had 5 years, and this patient had been given not 1, 7 antibiotics, maybe 2 at the same time, or 3 or 4, just to eliminate these bacteria from the lungs. It's a lung infection. And as you see here, the black dots is the bacteria. So even though you have this treatment, and the patient has received this treatment, you actually still culture the bacteria when they come to the hospital and give you a sample. And these bacteria are not resistant. So the questions that doctors are always asking is what's going on? The immunity, it's not super compromised, so they still have some part of immunity working. And that's where we decided to look at these persister bacteria. So bacteria that appear all the time, but we don't know why. And here is just another graph showing that on these 7 antibiotics that are given, that the majority of the isolates that we get from the patients are sensitive. So they should die in theory when you give this antibiotic. So coming back to the first slide, if you remember these guys, so we wanted to see how about whether these guys are the problem, why we see this. And these persisters and tolerant bacteria have been observed in 1944 by an Irish doctor, Joseph Bigger. And even though he saw Staph aureus persisters, Staphylococcus causes infections in skin or throat, this topic was neglected for centuries. And if you remember the slide with antibiotics, that's where the antibiotics were coming, the antibiotic-resistant bacteria isolated. So that was the focus of scientists. It was not these persisters. And then 10 years ago, people were like, okay, maybe we should look at persisters. So now there is a solid evidence coming that persisters are important, and they're trying to figure out what they do. Why do they sleep, or why are they so happy like these tolerant guys right here? So I worked with 40 patients, more than 500 isolates, and in the clinic, in the cystic fibrosis clinic. And basically, when I entered the lab, they had a lot of information on these 500 isolates, right? They had information about their genome. So what's hidden there, can we figure something out? And they were looking at resistance, at other phenotypes, and I was like, I will take this and then see how does it correlate with persister infection in these patients. And these colors here, they just present that something very interesting and something that usually people are not familiar with, and I would like to also show it tonight. And that's that when you hear Pseudomonas and when you hear these 40 patients, these patients are in the Pseudomonas in these patients is different. It's genetically different and phenotypically different. So we are genetically same, but we look different. They are also different a little bit genetically, but also phenotypically. And I'd like to show you a plate from the lab. So this is all Pseudomonas from the different patients. Some are a little bit red, some are a little bit green, some are smaller, some are bigger. And this is what happens in the clinic. When you get a bacteria and you say this is Pseudomonas, it's not the same for every patient. And that's why personalized medicine is coming now to be very important thing. And these patients have started from last summer to do this, to have this personalized treatment, that you would look at the color of the bacteria, look at the genome, and then decide what to treat them with. Okay, so this is just a small side story. So other thing from the lab, it's of course a lot of plates. So how do I know what is persister, right? When I take the bacteria from the patient. So there is something that we have developed, and that's you plate bacteria, you dilute them, and you have tons, in some place 10, in some 5. So then based on how many you get, which is the number here, and based on generations, this can be time or whatever, you have these kind of graphs and curves. And these curves are basically telling you what kind of action of the antibiotic is on that bacteria and whether you have resistant or non-resistant or persistent bacteria. And this is really nice, and we know it, so we can use it in the lab. So the situation with the persisters is that we have population of bacteria that we start with. We take the sample from the patient, we add antibiotic, and then you kill the sensitive to the antibiotic population, so you decrease the number of bacteria that survive, and then you end up in this kind of plateau curve here with red bacteria, which are the persister cells. If you take out the antibiotic, you get this curve again, and this curve again. So you cannot kill them, because once you take out the antibiotic, they will grow to a normal population. But when you have the antibiotic, you will get these guys sitting and not doing anything. Whether they're alive, whether they breathe, whether they produce some metabolites, that's something that we currently investigate in order to characterize, because once we characterize, we might develop a new antibiotic, you never know. But usually when you have something that is just sleeping and not doing anything, it's a little bit difficult to know how to attack them. So they do not grow, as I said, and then they can do this cycle on a very high concentration of antibiotics. So you can give them tons of antibiotics, they'll have the same behavior. So that's how in the lab I know that the bacteria is a persistent bacteria. And then we basically took the 500, you have these very nice plates that we can work with small volumes. I add a lot of antibiotic, in this case it's ciprofloxacin, and then this is without antibiotic. You see all the dots, they say that the bacteria grew fine, and then you have blank spaces where you say that you killed the bacteria, and then these guys here, they grow, which means that they are the persisters. And of course we do a little bit of more validation, and then you number them, and you end up with, in the collection of 550, 25% show this persister phenotype, and that is a little bit concerning. We have the whole genome sequencing, and let me know how many time.
Good.
And then, okay, so we do whole genome sequencing, which means it means that we know all the genes in every single 500 isolate, what it does, whether it's mutated, whether it's not mutated. And you can use this information to say, are they also genetically different, these persisters? Can we learn something from that? So basically, what you can do is take an isolate from the same patient early, like stage year 1, and then you go in year 2 and you take another isolate, and then you compare them and you fish for differences. So basically you'll end up with a table of genes that are mutated in this guy but not mutated here, and vice versa. And you can go in the lab, manipulate these genes, cut it off by CRISPR or induce new mutations, and then figure out whether that mutation is very important for this bacteria to be persistent. So we do this in multiple cases and we compare them. So basically this is a whole genome, and when you fish out only those differences of the genes, this is how it looks. This is one bacterial strain, this is another, and then the tree too are a number of different kinds of mutations. When we did this, it was so complicated and they're so various that you cannot say one gene is one function and so forth. So people kind of investigate all kinds of things using genetics, but it's very important to do killing curves and further characterize. So the genetics, it was important, we show, but not to a very high extent. Back to the patients, kind of I took this slide as a final slide of the study, and that showed, so the black curve, it's how many patients, when you add them, that have these high persistor isolates, how does that look like? And this is the time of colonization, which means that we had a patient infected at time 0, and we had followed them for 7.5 years. So we had already stored isolates with this length of infection. And we saw that in the majority of the patients, the longer time they were infected with the bacteria, the higher the chance that that bacteria would be a persistor. So we showed this incident, and it is concerning, but whether it's a causative effect or of what we observe, or there are other effects that contribute to do, it's still I would say a question which we always discuss and debate in the group. And as the last slide on drug resistance, because it's still there, drug resistant, it still appears, and once you have a resistant bacteria, what happens to the body and how is this linked to what we see as persistor? Because as you saw from the first picture, all the three are an interplay, so you can have all of the three types of bacteria at the same time. And how do one contribute to the presence of the other? This is just an illustration that if you have bacteria and you add antibiotic, then you can decrease the load. Once you remove the antibiotic, they grow again. Once you add the antibiotic, they die again, and so forth in many cycles. So this happens in different kinds of treatments. That's actually what happens in real life when you take antibiotics often. And then by the time, if enough time given, you end up with straight line. These are the resistant bacteria, and inside here you will have some of your persistor bacteria. And the idea and what scientists believe right now is having a resistant bacteria gives a higher chance that the persistor will come in a population. So these guys are also bad, so we need to definitely eliminate and put more money in research for drugs development. But we should not forget persisters, right? So in the end of the day, I think I would just end up on a slide, keep an eye on persisters for those that do some similar research or go to a doctor and get treatment for something and then the bacteria comes again. Keep an eye on these guys because they might be the cause of what's going on. So yeah, that's all. Thank you for the attention. Yeah.
Thanks very much, Bibi. If anyone has any questions, please do ask her. Like, what's an isolate? What's CRISPR? Which one is the baddest guy? I don't know. Just go ahead and ask.
How do you how does the resistance of the bacteria in the population develops? So you observe the persisters. Is it more like phenotypic thing that you have, for example, a population of bacteria that have some sort of different phenotypes, but they're genetically the same, and then you treat with a drug, and this certain phenotype stays, or are they genetically different?
Yeah. So.
Or do you induce different phenotypes by treating with drugs, and that's how the persisters develop?
Yes. So these guys here, specifically, right? When you start with a new population, you can add antibiotics, And in most of the cases, different antibiotics will give you different types of persisters, depending on how they kill, what's the mechanism of killing. So those are induced persisters. Resistant guys, you just add a lot here, add rock, and you don't have this kill, you basically have a straight line. So you know them exactly how they look. Phenotypically, in this, if I go back to this picture here, you cannot know. Like I tried to see whether they have a phenotypic trait, whether they're all red producers or green producers. You don't know, they're all kinds. And persisters are stochastic. So they just happen to appear in the moment and then they just regrow to normal population. That's why I've met many scientists that don't even believe that they exist, right?
So it's like you treat
Yeah.
population with a drug,
yes.
then you have persisters, and then they repopulate the whole population that is still sensitive to the same drug.
Exactly, because these guys will grow up to be this population once the treatment is over. And that's why it's so difficult to isolate and work on these guys. You can do it, and genetically they're the same. They're all the same genetically. It's just that they want to sleep, they don't want to sleep. Like it's very complicated mechanistic studies we do right
It's
now.
like it's like cancer.
But
I work in cancer.
Yes, they're also tolerant, right?
Yeah,
Tolerant cancer cells.
Sort of
Yes.
very similar, and I wasn't aware that you observe the same thing in bacteria.
And it's even more difficult because they're so dividing fast that you cannot like treat it or find a way how to stop this to form. But yeah, you're right. Yeah.
Any more questions? Come on, don't be shy. Okay, so I think it's beer o'clock now, and if you have any more questions for Bibi, just ask her. The kitchen is still open until 8? Until 8:30. So if you want to get something yummy, go ahead, or some more beers. So we'll do like a 10-15 minute break, and then we'll continue with one more talk. Thanks more, Sarah.
Ha ha ha.